Hi class, we're going to talk about the plague as we were working through diseases of the cardiovascular system. The plague has been around for quite a while. It's been occurred since antiquity. A pandemic of the plague has been occurring periodically as long as we have recorded history. The most notable outbreak of the plague was probably the occurrence that occurred in medieval Europe with the Black Death. This killed an estimated 100 million people. Oh, whoa, what just happened there? There we go. It killed an estimated 100 million people and actually caused the overall world population to decrease during that period of time. The signs and symptoms of the pneumatic plague are a respiratory disease, which we're going to discuss in more detail in chapter 19. However, we have the bubonic plague. This is another form of the same disease. Bubonic plague is caused when we have the bacterium that causes this disease, the plague, get into the victim via the bite of a flea. This bacterium, Yersinia pestis, then travels through the lymphatic vessels to the lymph nodes and then causes inflammation and necrosis of the lymph node. The lymph node will swell and cause a lesion known as a bulbo. These bulbos are usually going to form in the groin or the armpit areas. Occasionally they'll also last in the neck, form in the neck or the abdominal region as well. The incubation period between exposure and the development of the symptoms is between 2 to 8 days, and usually is going to culminate with fevers, chills, headache, all your general malaise symptoms. But the big symptom that's going to be associated with this is the tenderness of a bulbo. There'll be a dramatic swelling of a lymph node that is quite tender. The mortality rates for bubonic or bubonic plague are greater than 15%. There's also septicemic plague. If we take that pathogen and introduce it to the bloodstream, we'll then have septicemic plague develop. The pneumonic and bubonic plague are typically going to result in septicemic plague. Eventually, that bacterium makes its way into the bloodstream. This presence of the bacteria in the bloodstream causes widespread intravascular coagulation. DIC, or DIC, disseminated intravascular coagulation. So we're going to have widespread blood clotting all over the body. We're also going to have subcutaneous hemorrhaging or purpura forming. These are going to oftentimes degenerate and form gangrene. Mortality rates of septicemic plague are quite high, 30 to 50% with treatment and 100% without treatment. The main symptom of septicemic plague is darkening of the skin, and this darkening of the skin is why we call this disease the Black Death. Here is the telltale darkening of the skin that's associated with septicemic plague. Here is a swollen lymph node that's from the bubonic plague. And then the pneumonic plague is when the infection occurs in the lungs and fills the lungs with fluid. So here we can see day 2 of the symptoms. The patient's lungs are almost 2/3 filled with fluid, but this patient recovered, and by day 18, their lungs were almost cleared. So the plague is caused by Yersinia pestis, or Y. pestis. This is a bacterium that's very tiny. It's gram-negative. It's also a rod-shaped bacterium. It's a member of the bacterial family known as Enterobacteriaceae. This bacterium is going to display an unusual bipolar staining. So it's going to stain. When we stain this bacterium, it's not going to stain evenly. One side of the bacterium preferentially is going to absorb the stain compared to the other side. This bipolar staining of the bacterium gives it the appearance of a safety pin when stained underneath the microscope. Generally speaking, the number of bacterium needed to initiate full-fledged infection can be as low as 3 bacterium getting into the body, or 50. This is one of the most pathogenic or virulent bacterium known to humankind. Generally speaking, fleas are going to be the principal agent of transmitting this bacterium from organism to organism. And if we have more fleas, we can have more cases of the plague. The fleas, generally speaking, are going to be transmitted on an animal vector, such as a rat. After the flea ingests a blood meal from an infected vector, like a rat or a human being, that infected animal, the Yersinia pestis will rapidly multiply within the gut of the flea, and then the flea's esophagus becomes blocked because of the coagulation factors produced by Yersinia pestis. The flea is then unable to feed, it's unable to eat, and it's going to very rapidly jump around and in a panic, so to speak, try to get some food into its body by jumping around from animal to animal, from potential host to potential host, biting all these potential hosts and regurgitating infectious material into the bite wounds of the potential hosts. The plague is distributed across the planet right now. It's been reduced in incidence worldwide, but it's still present worldwide. It's currently increasing in incidence in Africa right now and a few other selected parts of the world. If we look at the United States, it's endemic, particularly in the western and southwestern states. We have an endemic plague problem. In the United States, annually there's a very small percentage of the population, or a very small number of cases, I should say, of plague that still are going to develop. Generally speaking, the people that are at risk for developing the plague are going to be veterinarians, or people that spend a lot of time living or working in woodlands and forests and with lots of animals. That incubation Another related disease is Tularemia. Tularemia has some various specific signs and symptoms, including backache, chills, and fevers. Depending on the immune system of the patient and how the portal of entry for the body, Tularemia can have an incubation period as short as a few days to as long as 3 weeks. Depending on how the pathogen enters the body, Tularemia can result in ulcerative skin lesions, swollen lymph glands, sore throat, intestinal disruption, pulmonary involvement. Tularemia, when treated, has a death rate or mortality rate of 30% within the United States. The pathogen that causes Tularemia is Francisella tularensis. F. tularensis is a facultative intracellular gram-negative bacterium. This is a pathogen that can live inside of, or bacterium that can live inside of host cells. This bacterium has a couple characteristics in common with Yersinia pestis, the cause of the Black Plague. Both of these bacterium are have been previously grouped in the genus Pasteurelliae. Tularemia is a disease that we contract from animal vectors. It's a zoonotic disease. The animals that are associated with are mammals that are endemic in the northern hemisphere. Sometimes this disease is also known as rabbit fever. As individuals would trap and skin rabbits, that used to be the most common method of this bacterium getting into the body. Right now, Tularemia is listed as a pathogen of concern for potential bioterrorism. It's potentially going to be used as a bioterroristic pathogen. Rabbits and rodents used to be the most common source of infection and are still a chief reservoir. We can also have other small mammals that can serve as a reservoir as well. Currently, the most common mode of transmission is going to be a tick bite. It used to be from handling rabbits, but since most individuals don't work with rabbits now, skinning and trapping rabbits is not a very common activity anymore. We've had an increase in tick bites and mosquito-borne vectors inoculating patients with this bacterium. This bacterium can cause an infection if we have as few as 10 to 50 microorganisms get into the patient. This is on par with the Yersinia pestis in terms of being one of the most infectious of all bacteria. Cases of Tularemia have been appearing in people who even occasionally run over dead rabbits with a lawnmower. In those situations, it's been assumed that the bacterium entered the patients via the respiratory tract as they inhaled aerosolized bacterium. Back in 2009, there was an incidence where 2 people in Alaska acquired this disease by wrestling infected rabbits from their dogs' mouths. It's quite easy to contract because it has such a high infectious rate. To prevent and treat Tularemia, we typically are going to have an issue with the fact that it can be maintained intracellularly. This bacterium can hide inside of our own cells, making it very difficult to treat and leading to frequent relapses. We can use antimicrobial chemotherapy or antibiotics, but we can't use antibiotics indefinitely, and at the same time, we can't stop using the antibiotics after the symptoms go away. So just because we've had the regular regimen of antibiotics doesn't necessarily mean that we should stop treatment. There needs to be a prolonged period of treatment, but not an indefinite period of treatment. We can typically get protection from a live attenuated vaccine. This vaccine is not very common because this disease is not very common. Individuals that are occupationally exposed to lots of animals should also wear gloves, masks, and eyewear, in addition to being preemptively vaccinated against this disease. If we look at Tularemia, it's caused by Francisella tularensis. To review, is commonly contained or transmitted via small mammals and or arthropods. And to prevent it, we should take the vaccine. For high-risk individuals, to treatment, we should take a prolonged regimen of antibiotics. And within the United States, we only have several hundred cases annually. Up next, we have Lyme's disease. Lyme's disease is an often non-fatal disease that evolves from a progressive syndrome. Lyme's disease can mimic many other diseases, such as neuromuscular and or rheumatoid arthritis, which helps or is one of the reasons why it's been misdiagnosed in many cases. It can also result in erythema migrans. This is also known as the bullseye rash. This is an early symptom in the majority of cases, approximately 70% of cases. This symptom is going to result in a red rash that's going to spread outward in concentric circles from the bite or the portal of entry. Some other symptoms that are associated with early infection are going to be fever, stiff neck, and or occasional dizziness. If not treated, or if it's treated too late after infection, this disease can oftentimes advance to the second stage. The second stage is much more profound and will cause cardiac and neurological symptoms, occasionally leading to facial palsies. And then there's the third stage. This third stage of the disease can lead to crippling arthritis in the patients and will oftentimes cause chronic neurological complications that are quite disabling to the patients. If treated early with antibiotics, there's a high success rate of recovery, but if it makes it to the second or third stage, the success rate of treatment diminishes drastically. Here is the classic bullseye rash manifesting itself on a patient. Lyme's disease is caused by Borrelia burgdorferi. This is a bacterium that's spirochetan shaped, and it's going to be quite large, between 0.2 and 0.5 microns or micrometers in diameter, and 10 to 20 microns in length. These bacterium are going to contain 3 to 10 loose coils, depending on the individual bacterium and how spiraled it is. This bacterium has some very unique nutritional requirements. They're quite complex and cause it to be very difficult to culture this bacterium within a laboratory setting. This is one of the reasons why it's so difficult to treat the disease in the secondary and tertiary stages and why it's such a difficult disease to specifically diagnose. The bullseye rash is typically the most common method of diagnosing this disease. This bacterium is very easily can evade our immune system. It can change its surface antigens. So while our immune system can develop an adaptive response to one form of the bacterium, this bacterium can then alter its surface proteins, alter its surface carbohydrates, so that our immune system can no longer have an adaptive response to it. And because it can constantly change its surface antigens, it makes it much more difficult for our body to fight it off with a typical immune response. This bacterium is going to give us a very strong humoral immune response or blood immune response. This blood response, though, is typically ineffective because the bacterium is constantly switching its surface antigens. And it's possible that this immune response is going to attribute potentially to the pathogenicity or pathology of the infection itself. As we have this highly elevated humoral response or blood-based immune response, it could potentially lead to some of the symptoms associated with this disease. Generally speaking, this disease is going to be transmitted by ticks within the genus Lyxodes. Lyxodes scapularis, the black-legged deer tick. This deer tick is found in the Northeast and is going to be capable of passing this disease. This disease this tick goes through a 2-year life cycle, and this tick is going to have 2 principal hosts. If we look at Lyxodes pacificus, this is the black-legged tick that's found in California. And that tick is primarily going to be found on a dusky wood rat species. Here in Minnesota, the most common form of the tick that we are going to expose to is Lyxodes scapularis, or the black-legged deer tick. And the 2 principal hosts are humans and deer. The greatest concentrations of Lyme disease are found in areas that have high deer populations, because high deer populations are associated with the black-legged deer tick. If we look, we can see that we have the black-legged deer tick infecting the deer. From the deer, that tick can go to a mouse, and then from the mouse, that tick can go to either a human and or a deer. Generally speaking, the best way to avoid getting this pathogen is to minimize your exposure to areas that have lots of deer and or mice. If we look at a map showing the distribution of the disease, this disease is found primarily on the East Coast and in Minnesota and Wisconsin within our United States. Because Lyme's disease, because the bacterium has such specific nutritional requirements, it's very difficult to culture and diagnose this disease. The most suggestive sign is that bullseye rash, or that ring-shaped lesion. But if we can isolate the spirochetes from the blood of the patient, that typically is going to be another symptom that's indicative of the disease. We can also use ELISA testing to test for the antibodies that are associated with the pathogen. Tests for spirochete GNA are also increasing in frequency, particularly in the late stage diagnosis after the infection has been altering its surface antigens for an extended period of time. For a very brief period of time, there was a vaccine available that could prevent Lyme's disease, but it's been removed from the market. Anyone right now that is involved in outdoor activities, particularly in areas that have high deer populations, should avoid exposure to the tick. So wear protective clothing, boots, socks, and some insect repellent as well. These are all ways to keep the ticks off our body. If you're exposed to a heavily infested area, you should also check your body regularly for ticks. When removing the tick from the body, do it without crushing the tick. So use some small forceps or tweezers and gloves, because it's possible to become infected by this tick feces or the body fluids of the tick as it's being removed from the body. If diagnosed early, there should be a prolonged treatment of 3 to 4 weeks with doxycycline and amoxicillin, and that has been found to be effective at keeping that disease from entering the secondary or tertiary phases. To review on Lyme's disease, it's caused by Borrelia burgdorferi. It's going to be transmitted primarily by the deer tick. This disease can be diagnosed via some immunoglobulin or immunologic methods. The best way to avoid it is not get bitten by a tick. And within the United States, there are approximately 300,000 new cases a year, mostly aggregated on the East Coast and Minnesota and Wisconsin. So, concept check class. Which of the following diseases still appear within the United States? It's the plague, Tularemia, Lyme disease, or none of the above. Go ahead and pause this video, check your notes of the PowerPoints, or rewind the video and get me an answer. 5, 4, 3, 2, 1. The correct answer is A, B, and C. All three of those diseases still occur in the United States, although of those diseases, by far the most common is going to be Lyme's disease. If you have any questions about this material, please feel free to post them to the discussion board or shoot me an email. Happy